Welcome friends, myself Shoptapurni from BioLive. Hope all of you are doing very well. Today I am going to teach you about next generation sequencing. So nowadays you have heard about the term next generation sequencing. It is very popular nowadays. Why? Because see here, in Sanger sequencing you can only sequence a short stretch of DNA at a time. But in case of next generation sequencing, you can sequence the whole genome at a go by the fragmentation method. Okay, you have to just fragment the whole genomic DNA and you can assess the sequence of each fragment at the same time. That means you can assess the sequence of the whole genome at a go. So that means it is less time consuming. Second one, it is cost effective, and third one, obviously the error in this method is also less. Okay, it is almost 98% correct. So for these three characteristics, next generation sequencing is so much popular nowadays. So before beginning, I would like to request you to subscribe to my channel. If you like my video, don't forget to hit the like button, and if you find it useful, don't forget to share it with your friends. And if you have any doubts, you may leave a comment below in the comment section. You can also follow me in my Facebook page, Facebook group, or in my Instagram profile. The links are given in the description box below. So okay, let's begin. See, what is the basic principle behind the next generation sequencing and what are the different steps of this kind of sequencing? There are actually 4 steps. What are those? The sample preparation, cluster generation, sequencing, and the fourth, last but not the least, the data analysis. Okay, so see how you will prepare your sample. First of all, you have to isolate the genomic DNA from your sample. Okay, and after isolating the genomic DNA, you have to amplify that by the process of PCR. After amplifying the genomic DNA, you have to make it into fragments by the process of fragmentation. See, it is our whole genomic DNA and you have made it into different fragments by the process fragmentation. Okay. Each fragment is destined to be bound to each flow cell in the glass slide. Okay. So to bind these fragments to the flow cell, you have to attach some adapters here. Adapters are actually genetically engineered oligonucleotides. Okay. See here, we have added 2 different types of adapters, green adapter and red adapter, to each end of your DNA template fragment. And each adapter has 3 different sites. Obviously, there will be a site which will be complementary to the oligonucleotide present in the flow cell. See here, these are the 3 flow cells present in the glass slide. There are numerous flow cells present in the glass slide, but I have only drawn 3 here. And each flow cell is coated with 2 different types of oligonucleotides, the red one and the green one. Okay, and these oligonucleotides are complementary to these adapters. Okay, so these adapters are actually added for hybridizing the template DNA strand with this flow cell. Okay, for the purpose of next generation sequencing. So obviously there will be a region in your adapter which will be complementary to these oligonucleotides so that they too can bind with each other. And there will be a unique index for each fragment. Okay, and these unique indices are needed for recognizing your fragments during the data analysis method. Okay, and obviously there will be a site for sequence primer binding. Okay, so these are the 3 regions of any kind of adapter. So now coming to the second step, the cluster generation. For cluster generation, you have to take a glass slide on which you will find numerous flow cells like these ones. Okay, and each flow cell is coated with 2 different types of oligonucleotides, and these oligonucleotides are complementary to these adapters. Okay, so for cluster generation, first of all, you have to denature this DNA fragment. Okay, and after denaturing it, one of the strand will bind or hybridize with the complementary oligonucleotide here. It is our template DNA strand which have hybridized with the red oligonucleotide here. After hybridizing, the DNA polymerase will be recruited here and the complementary strand will be generated here. Okay, see this is the complementary strand. After generating the complementary strand, the original template strand should be washed off. I have drawn the arrow here to show that the original strand is being washed off. Okay, So now there is only the complementary strand, one complementary strand in your flow cell. So now comes the main part of next generation sequencing known as bridge amplification. In bridge amplification, the newly synthesized strand will hybridize with the other oligonucleotide present in the flow cell. Okay, that means this new strand will now hybridize with the green oligonucleotide present in the flow cell, and this will form a bridge-like structure. Along this bridge, the new strand will be synthesized from this green oligonucleotide. See here, this is the new strand to be synthesized. Okay, and this kind of amplification is known as bridge amplification. So after this process, you will find both the forward and reverse strand of your template DNA present in your flow cell. Okay, this is the forward strand and this is the reverse strand. And obviously they will now form the bridge with their complementary oligonucleotides present in the flow cell. See here, this one will form a bridge with the green oligonucleotide, and this one will form a bridge with the red oligonucleotide, and that is how they will repeat the cycle and will form a cluster of forward and reverse strand in each flow cell. Okay, see here, in this cluster you will find both forward strand and reverse strand are present here. Okay, and this kind of amplification by bridge amplification is known as clonal amplification. Okay, so now you want to know the sequence of forward strand. So for that, first of all, you have to remove the reverse strand from the cluster, okay, from the flow cell. So these are the reverse strand. Okay, so I have drawn the arrow sign to show that these reverse strand are being washed off from the cluster. So only now forward strands are present in your flow cell. So these forward strand can obviously bind to the their complementary oligonucleotides in the flow cell and can again start the bridge amplification. To stop that, to prevent that, you have to block the 3' OH end of the forward strand. Okay, see here, I have drawn a line here to show that the 3' OH ends of the forward strands are blocked, so that bridge amplification cannot occur again. Okay, after blocking the strand, now the third step comes, the sequencing step. See here, it is our forward strand, okay, and the 3' OH end is blocked, so that it can remain in this configuration, so that the sequencing can be done. Okay, now you have to attach the sequencing primer. The small red one is our sequencing primer. After attaching the sequencing primer, you have to attach different types of DNTPs which are labeled with fluorescent probes. Okay, see here, the ATGC, DATP, DTTP, DGTP and DCTP are labeled with different color-coded probes. Okay, that means whenever the complementary DNTP will be added here, you have to administer a laser light to it and it will glow up, okay, with different colors. Like if T is attached, it will glow up in green color. If A is attached, it will glow up in red. If G is attached, it will glow up in blue, and C is attached, it will glow up in yellow. Okay, that means whenever the DNTPs will be added, it will glow up with different colors. And see here, obviously during this time, obviously let's say here the fragment 1 is bound. Okay, here the fragment 2 is bound. In this flow cell, fragment 3 is bound. Okay, obviously at the same time, in each of the fragment, the first DNTP will be added, and let's say for the fragment 1 it is T. So this flow cell will show the green color. Okay, for the fragment 2 it is A. So this flow cell will show the red color. For fragment 3 the first DNTP is C. So this flow cell will show the yellow color. So at the same time, different flow cell will show different color. Okay, as different DNTPs are added there. So as it is, the cycle will run and it will show different colors at different flow cells, and by assessing those colors you can find the different DNTPs, order of DNTPs in your template fragment, template forward strand fragment. Okay, so now you have found the DNTP sequence in the forward strand, and if you now want to sequence the reverse strand, okay, to to confirm your sequence actually, then what you should do? For that, what you should do, first of all, the newly synthesized strand should be washed off. Okay. And after washing off the newly synthesized strand, you have to remove this 3' OH block, okay, OH end block, so that this forward strand can now again form the bridge. See here, the forward strand has again formed the bridge, and along this bridge, the bridge amplification will occur. And after that, the forward strand should be washed off and only the reverse strand should remain here. See here, only reverse strand is remain, and the reverse strand, for the sequencing of the reverse strand, you have to attach the reverse strand sequence primer, green one here, and then the DNTP should be added. And see here, this sequence is exactly opposite to this sequence, reverse to this sequence. Okay, so similarly, like the forward strand, new DNTPs will be added here, and each flow cell will show the different colors, different fluorescent colors here. Okay, and that is how, like the forward strand, reverse strand can also be synthesized. Okay, and next we will see how from sequences of different fragments you can generate the whole genome sequence. Okay, from the library of different fragments and their sequences, their forward and both the reverse sequence, how can you generate the sequence of the whole genome? Okay, by the method of data analysis, which is done in the bioinformatics softwares or tools. Okay, so our fourth step of next generation sequencing is data analysis. So for data analysis, you have to find out the overlapping sequences of different fragments. Okay, and according to that overlapping sequence, you have to arrange those fragments, and after arranging those fragments, you can find out the sequence of your required genome. Okay, so see here, we have arranged the three fragments, F1, F2 and F3. We are just taking the fragments from 3 flow cells because it is not possible to show all the fragments in our whiteboard. Okay, so we are just taking 3 fragments, and for each fragment we have taken the forward strand and reverse strand sequence. Okay, so these are the forward strand sequences for 3 different fragments, and these are the reverse complementary sequence, strand sequence of the reverse strand. Okay, these 3. Okay, and after seeing this, it is clear that there are some places which is common to both the fragments. Okay, and those are the overlapping places or the matching place. Here AT is the overlapping place of F1 and F2 fragment, and GA is the overlapping space of F2 and F3 fragment. Okay, so after observing the overlapping sequences, we can arrange these 3 fragments like this, and from this you can easily detect what is the sequence of your fragment. How? A, T, C, G, A, T, then G, C, T, G, then A, T, T, A, G, C. This is the required sequence of your genome. Okay, and that is how you can find out the whole genome sequence by arranging all the fragments by their forward and reverse strand. Okay, so this is all about the next generation sequencing method. And if you have any doubts, don't forget to leave a comment below in the comment section. I hope now the concept is crystal clear to you. So I will see you in the next video with another interesting topic. For the time being, thanks for watching and happy learning.